Well, it's a real pleasure to be here. Let me what I'd like to do, as you can see from the title, is combined systems medicine, stem cells, and space. That's what I was asked to talk about, and that is not a trivial set of connections, but let's see exactly how I do. So systems medicine is really this systems approach to biological complexity that resides in disease. And I'm going to talk about exactly what systems medicine is and give you some examples of what we're doing with systems medicine. At the very end, we'll talk about a stem cell example, but throughout, we'll talk about how these technologies and strategies could conceivably be applied to space. So I would say systems medicine really has two cardinal features. One, it is about big data. The way you deal with biological complexity is to create big data. And of course, by big data, we mean data that ranges from molecular and cellular all the way up to the social networks of individuals. There are two challenges with big data. One is how you do the integration of these data to create for each individual patient a model that optimizes their wellness and minimizes their disease, and we'll talk more about how you can begin to do that later. And the second challenge is in any type of big data, there are enormous signal-to-noise problems, and I'll talk in a few minutes about how we deal with that. One of the ways to take the billions of data points that will surround every patient in 5 to 10 years and reduce them to a dimensionality that gives us insights into disease is to use them to decipher what we call the network of networks. So biological networks operate at the chromosomal, the molecular, the cellular, the organ, and finally the individual levels. In an individual, they're seamlessly integrated. Those networks manage information. When they become disease perturbed, the information content alters. If you can capture that information content, which we can now, that altered information content, you can make develop fundamental insights into disease mechanisms and think about new strategies for approaching diagnosis and therapy. Now, the essence of how systems medicine thinks about big data is, one, it should be as global as possible. It's possible to be global with genomic types of data in general. It is very difficult to be global with the other types of data. We don't have the tools to do comprehensive analyses, although as you'll see, that's starting to change. It's key to do the dynamics of systems. The most important, perhaps single way of dealing with signal-to-noise issues is to understand the dynamics of disease. And of course, that's virtually impossible to do in humans, so one really has to think about animal models. You have to be able to integrate data to make the models that we talked about earlier. But what is really critical, again, is dealing with noise. And the noise is of two types. There's biological noise. There's technical noise that arises from the nature of measurements and how you handle them. And I'll give you an example of that in the next example we'll talk about, which is one example of disease dynamics we've studied in a mouse model system where we induced neurodegeneration by the injection of infectious prion particles into the brain. And in doing so, we created a disease in mice that lasts over a period of 22 weeks. We interrogated the brain transcriptomes at 10 different time points across this dynamical time period, subtracting at each time point a normal transcriptome from the disease transcriptome counterpart to identify the genes that are differentially expressed. And of course, we were horrified to see that about a third of the mouse genes were expressed in the brain differentially. And that means there's an enormous signal-to-noise problem. So we created, and the major source of noise is biological noise. It's the other biologies than neurodegeneration that contribute to changes in the brain transcriptome. We created 6 inbred prion strains of mice that allowed us to subtract away major aspects of biological noise. We ended up with 300 genes that we thought reflected neurodegeneration. And what we then did was to look dynamically at the histopathology of this disease and demonstrate that there were 4 major processes operating: prion replication and accumulation, glial activation, and 2 forms of neurodegeneration. And we mapped onto each of those 4 processes these differentially expressed 300 genes into protein-protein interaction networks, and came to 2 major kinds of conclusions. One, that the networks, the 4 major networks were expressed, became disease-perturbed in a sequential manner, starting with the most specific network, that is prion replication and accumulation. And this sequential disease perturbation of networks is key, because if you're interested in diagnosis or therapy, it's the most proximal network you'd really like to look at and come to understand how to convert back to normal before downstream networks are converted into the disease process. The second point we realized was only 200 of the 300 differentially expressed genes mapped into those 4 networks. The other 100 defined 6 additional smaller disease-perturbed networks that no one had any idea were a part of this disease process. Collectively, the dynamics of these 10 networks explain virtually every aspect of the pathophysiology of disease. So you can see the power of looking at dynamics in the mouse model. Now, we're also interested in a variety of technologies, and I'm only going to highlight them very quickly. 3rd-generation sequencing, single molecule nanopore electronic detection is absolutely going to be revelatory. It is going to enormously increase the throughput. I see in a 5 to 10 year period doing a human genome in 15 minutes at a cost that will be under $100 per genome. So there's going to be a real revolution in genomics. And with some of the genomic strategies that are being suggested, we'll be able to get the epigenetic marks on DNA and to read 200 kb DNA fragments, so we can do de novo assembly of the sequences independent of prototype sequences, which are always flawed and error. So 3rd-generation sequencing is going to lead a revolution. My prediction is in a 5 to 10 year period, you'll all have your genomes as a part of your traditional medical record. I think there are a variety of other possibilities that we can talk about too. Proteomics is going to become global with a new type of swath proteomics that ISB and other people are pioneering. Targeted proteomics now give us the ability to look at 100 proteins in an hour using mass spectrometry at a mid-atomole level, even in complex mixtures. We're working on procedures to get antibody capture agents that will replace antibodies, that will be more stable and digital in nature and more highly focused and targeted at particular epitopes, so you eliminate cross-reactivity. And my belief is these peptide protein capture agents will replace monoclonal antibodies in the next 10 to 15 years. Single-cell analysis I'll talk about in a few moments. I won't say any more about that, and I'll mention briefly some of the types of analytic tools. What about system strategies for doing medicine? Family genome sequencing integrates genomics and genetics, lets us readily identify disease genes. We've now developed systems approaches to blood diagnostics that make creating diagnostic protein and or microRNA panels absolutely routine if you have good phenotypic distinctions. I'll talk about a longitudinal assessment of wellness using a Framingham-like model for examining 100,000 well patients. And then we'll end talking about pluripotential cells and how we can use those to study development and so forth. Family genome sequencing with this integration of genetics and genomics not only makes it easy to find disease gene candidates, but it gives us the ability to reduce sequencing errors, to be able to identify rare variants, and most important, to determine for each member of the family precisely their chromosomal haplotypes. That's important because you can ask the question, what fragment of the haplotypes are shared by the disease individuals that are not seen in the normal individuals, and you can reduce enormously the chromosomal search space for disease genes. We were able to use this technique to reduce the candidate number to 4 genes, and it was relatively simple to assign a gene for each of the 2 disorders that the children actually exhibited. And again, using family genomics, this shows the power of being able to identify disease genes. Here we looked at 5 families, and one of the families actually had a gene that had been discovered before, the ATP1A3 gene, as well as 15 out of 18 sporadic individuals. But the other 4 families each exhibited a disease gene, one shared by 2 families. And in each case, the family was sufficient to define the disease gene because of the principles that we've talked about. So I think one of the interesting possibilities then for thinking about space is, are there people that are genetically more fit to be able to survive in space? And of course, that gets down to, can you define sharp phenotypes where we can do the analyses? How do we do blood diagnosis using a systems approach to make blood a window into health and disease? Of course, blood is ideal because it bathes all organs. They secrete proteins into the blood. Those proteins report back on the status of the organ. Hence, if you knew the location of proteins you were looking at and studied them in a longitudinal sense, you could access the status of that organ and so forth. And I'll show you how we can actually identify a subset of these molecules by defining them as organ-specific in just a few moments. The real pioneering breakthrough for doing blood protein diagnostics is targeted mass spectrometry, which, as I said, allows you to analyze 100 proteins in an hour at the mid-atomole level, and you can quantify these proteins very nicely with isotopic peptides. What ISB has done in the last 3 years is actually created targeted proteomic assays, SRM assays, for virtually every human protein. So we have an atlas that's essentially a lookup book where we can get the assays for any set of 200 candidate proteins that we'd like to examine in the context of a particular diagnostic panel. We've actually done deep transcriptome analyses of the order of 50 organs from humans and from mice, comparing the transcriptome of each organ against all other organs to identify transcripts that are organ-specific. And then we've asked the question, do those organ-specific transcripts secrete proteins into the blood or shed proteins into the blood? And in all cases, we've been able to find reasonable numbers of proteins, some for more complex organs being large in number, like the brain. We have almost 200 organ-specific proteins. So the idea is, if you have a normal brain and look at it longitudinally, the 200 proteins will be set and one set of levels for a given individual. If that brain becomes diseased, a subset of networks become disease-perturbed that alters the concentration of their cognate proteins in a way that's characteristic for each disease, because each different disease has different combinations of disease-perturbed networks. Moreover, these 200 proteins all map back to generally known functions in the brain. So not only can you do pattern recognition to distinguish health from disease, and if disease, which disease, you can also make inferences about functions that might become disease-perturbed as well. And indeed, we took 15 of these proteins for the mouse with prion disease that mapped into each of the 4 major organs and demonstrated by doing longitudinal blood analyses 2 important things. One, very early diagnosis. We saw changes in the blood 10 weeks before you could even remotely see the first clinical signs. And number 2, we were able to show successively in the appropriate time period the disease perturbation of each of these 4 networks. So we can follow the progression of the disease in this state. So let me give you a solid example of what we've done then in humans, where we've got a protein blood diagnostic test that distinguishes benign from neoplastic nodules. And we did this with a company I started 5 years ago called Integrated Diagnostics. So the issue is there are 3 million lung nodules a year. As a pulmonary oncologist, you say, what do I do with this lung nodule? And the answer is you have a series of choices. You can have watchful waiting, or you can have immediate surgical removal, or you can have 600,000 in this dilemma zone where you'll do PET scanning or bronchoscopy or needle aspirations. And the unfortunate aspect is more than half the people that have these surgical procedures have benign nodules. So there's an enormous waste of money. There's an enormous cost in morbidity for normal patients. So the approach we took was to get a blood panel starting with 400 protein candidates, and we used systems approaches to choose the 400, and then a series of selective systems approaches to narrow the 400 to 13 to deal with signal to noise. And the panel of 13 is quite remarkable. It allows us to rule out unequivocally about 70% of the benign nodules, and we can calculate that this will save the healthcare system north of $3.5 billion a year and bring peace of mind to many patients. And in fact, this panel is just being commercialized as we speak. And what has been incredible is the response of the insurance companies to this panel, seeing economic benefits. So we now have coverage that approaches 100 million patients, and that's been done in just a few months. I don't think anybody has had that kind of remarkable progress. What is also interesting about the panel, because of the way we did the selection, the metrics are independent of the 3 classic criteria for making assessments about what to do with lung cancer, namely smoking history, age of the patient, and the size of the nodule. So here's a simple illustration of a set of patients, blue benign, red cancer, and it shows the 70% of the benign patients that we can rule out, that we can put in watchful waiting. There are a few, 10%, 20% with cancer, and the watchful waiting will identify those and can move them over into the surgical categories. What is really striking is you can ask the question, what do these 13 proteins represent? And in fact, what they represent is, well, 12 out of 13 map into 3 major disease-perturbed networks in lung cancer. So this filtering not only selected a panel of proteins that could do diagnostics, it gave you insights into the biology of the disease process that you're actually analyzing. So these panels can do then 5 things: rule out normal and neoplastic from bloods. They can do early diagnosis. We can follow the progression of the disease. And we will be able to follow the response to therapy. And really important, we'll be able to use these panels to stratify the disease into the distinct subgroups that will be necessary for appropriate impedance match against effective drugs. And I can talk a lot more about that, but we haven't time to talk about it. And of course, these are the kind of assays that you can use to look at diagnoses of osteoporosis, muscle wasting, any other physiologic phenomena that you'd like to study in space. A third strategy has to do with a new proposal we've made to have a Framingham-like study of 100,000 well patients over a 20 to 30-year period. And the idea is, in studying disease, we're a lot like the fellow looking for his lost coin under the lamppost, because there's light there, whereas he lost it a block away. When we study disease in humans, often we study the terminal end of disease. We do Alzheimer's studies where we look at the brains of dead people. We really want to know what Alzheimer's disease looks at the earliest onset of the disease. And we don't have simple ways to do that. So I'll show you a simple way for doing that. The idea then would be to take 100,000 patients and to follow them. A subset would either maintain wellness or would acquire even better wellness, and we can show you how we could do that. But another subset will show transitions from wellness into disease, and for 100,000 patients, we'll see significant samples of all major types of disease, cancer, cardiovascular, neurodegenerative, and the like. So the idea is, if we follow those patients longitudinally with multiple measurements of their features, then we'll be able to have enormous data on the earliest stages of disease transition. And what we'd like to be able to do is, starting with these well patients, is identify as early as possible the transition into disease and hopefully be able to move those patients back to a wellness trajectory. So what we propose to do is a complete genome sequence. What we propose to do is classic blood chemistry monitoring with an emphasis on nutrition 3 times a year, self-tracking with quantified self-measurements, the digital devices for respiration, heart rate. We even have some digital devices that'll do calories in, calories out, and things like that. And we are going to be looking at novel biomarkers. So, for example, we're going to be doing the gut microbiome 3 times a year. We're going to be looking at organ-specific blood proteins that will follow wellness to disease transitions for the brain, for the heart, and for the liver. And we'll collect all of these data and put it together in models for each individual patient that will allow them to optimize wellness and minimize disease. So we'll see transitions from wellness to greater wellness. We'll see transitions from wellness to disease, and we'll see transitions from disease to wellness, so we can study the dynamics of disease in humans in ways never before possible. And the idea then, of course, in the short term, is to be able to give each individual patient models that will optimize their wellness, minimize their disease. In the intermediate term, we'll be able to mine the data on the well patients for metrics of wellness, which are absolutely critical missing features in medicine today. In the longer term, we'll be able to take a database that follows the transitions from wellness to disease and to mine it for insights into the earliest features of disease, features that then can be subverted to move one back to wellness. And of course, with the databases that we'll generate from 100,000 patients, we hope to, in a sense, create a Silicon Valley of wellness and disease transition, an enormous amount of information that will, I think, be the vital opportunities to create a whole series of spin-off companies which are going to move into a wellness industry, an industry I think in 10 to 15 years will far exceed the disease industry, the classic healthcare industry. So we ought to be using these kind of assessments on astronauts on space and when they return back to Earth, so we can see not only about the perturbations that cause the effects, but about the responses that take them back to normal effects. So the last thing we'll talk about are the use of induced pluripotential cells to do biology and diagnostics. This is a company with Cellular Dynamics International, a stem cell company that has done a spectacular job industrializing stem cell biology. And I won't say more about it, but that's utterly essential to all I'm going to talk about. What got me interested in thinking about biology in new ways was observations we made about 5 years ago, taking 32 cells from a classic human glioblastoma cell line, analyzing 32 transcripts for each of those single cells quantitatively, and then being able to use those transcripts to map those cells into 3 discrete quantized states. Now, the question is, what are the quantized states? Are they successive stages of some neoplastic transformation, or are they different sets of cells that cooperate together to facilitate the neoplastic process? We can't answer that question, but where I really think it is relevant is looking at the differentiation of pluripotential stem cells to 1, cardiomyocytes, and 2, neurons. So in a very large scale, we're going to look at that differentiation process, generating enormous amounts of omic data across that process. But what we're going to do first is single-cell analysis across that process to identify the quantized states. Let me just say, identifying quantized states is really technically difficult because there's an enormous amount of noise in all the ordinary transcripts you look at. It's only a few molecules, generally transcription factors, that specify control of the quantized states. So looking at this transition of iPS to differentiated cells, we were particularly interested at the single cell level in analyzing transcription factors. So we plan across 30 days of this differentiation selection process to look at 8 different stages at the single cell level to identify the quantized states, to have cell surface molecules that will let us sort those populations, and then do deep omic analyses, genomic, proteomic, metabolomic, lipidomic, and the like on these, so we can generate a level of data that is absolutely incredible for development, but not be paralyzed by the existence of the complexity of living organisms in which development occurs. What we also plan to do is get individuals that have genetic defects in these differentiation processes. For example, patients with GATA4, a transcription factor, have a proclivity to go to cardiomyopathies, and we've actually done the complete genome sequence of most of the members in a 5-generation family that has such a cardiomyopathy. So what we'll do then is look, in that case, at a normal individual in the lowest family, the 5, 6, 7 families, and 2 diseased individuals to see how that gene is perturbed, the quantized cell states that lead to the development of cardiomyocytes. And we'll do the same for Huntington's gene to look at defects in the development of neurons as well. Here are a series of single-cell studies. So the x-axis is the 97 different genes, transcripts that we've quantified, and the y-axis are several hundred cells that we've looked at. So you can see here, looking at developed neurons and at the iPS cells, there are beautiful differentiations between those 2 cell populations. But when we looked in more detail at a fully differentiated set of neurons, we could show that they differentiated to GABAergic and glutamatergic neurons. That was the bulk of the cells. But a small fraction of those cells represented classic stem cells that hadn't changed. That's a part of all developed tissues. And another fraction represented stem cells that were at this branch point where they could go either way to the 2 types of neurons. And that gives us the ability then to begin looking at a lot of the physiologic details of maturation. So we can use these kinds of studies to assess how space residence may change stem cells in interesting ways. And the final point I'd make is systems medicine has really reached a tipping point in its changing the practice of healthcare. It gives us the ability to look at fundamental mechanisms, to identify disease genes in family studies, to create diagnostics that make blood a window for separating health from disease. I haven't talked about how we stratify diseases or how we stratify patients. They are really important. But a final point I'd like to make in this context is recently we carried out a series of studies on 100 individuals, or the blood from 100 individuals that had Alzheimer's disease and matched controls. And we were able to demonstrate unequivocally, not only were there profound changes in the brain, there were profound changes in the liver. And this raises the really interesting question of most diseases will be multi-organ, and how are we going to study those, that is, the organs that participate in the disease process and how they change together. And one of the key ways is going to be the organ-specific approach we've talked about. We've got new pioneering approaches to the identification of drug targets that I think will really revolutionize the time and cost of producing drugs, and we plan to do a lot of large-scale multi-parameter Framingham-like studies, and we've started some on preterm birth as well as cardiovascular disease, and I described the wellness assays. So I'd say systems medicine really provides powerful technologies and strategies for studying humans and cells and disease or wellness, and it's the marrying of these new kinds of technologies with unique opportunities that we're going to begin hearing from the panel that I think present enormously powerful opportunities, not only for science in space, but science on Earth. Thank you very much.